As we are aware, enzymes are larger biological molecules that are responsible for the hundreds and thousands of metabolic processes that maintain life. They are greatly exclusive catalysts, highly accelerating both specificity and rate of the metabolic reactions, from digesting food to synthesizing the DNA molecule. Most of the enzymes are proteins, and a few catalytic RNA molecules have also been found out. Enzymes adopt a particular three-dimensional structure, and they may be also utilize inorganic and organic components as cofactors to help in catalysis. In the enzymatic reactions, at the beginning of the process, the molecules called substrates are converted into some other different molecules, and these molecules are known as products. In order to occur at rates that are sufficient for life, all chemical reactions in a biological cell need enzymes. The enzymes that take part in DNA replication process are helicase, primase, DNA polymerase 1, 2 and 3, ligase, telomerase, nuclease, topoisomerase, single strand binding proteins and DNA gyrase. The main function of the helicase enzyme is to denature or unpack the DNA. Helicases are motor proteins. They move along a nucleic acid phosphodiester backbone directionally, separating the two strands of the DNA by using the energy that is derived from the ATP hydrolysis. Cellular processes like DNA replication, translation, transcription, DNA repair, ribosome biogenesis and recombination involves the separation of the nucleic acid strands that requires the use of helicase enzyme. Primase catalyzes the production of a short RNA fragment called a primer that is complementary to an single strand DNA template. Primase is very important in the DNA replication process because DNA polymerases cannot initiate the synthesis of a DNA strand without a DNA primer. Telomerase is a ribonucleoprotein having the RNA and a protein component in its structure. It adds the DNA sequence repeats to the 3' end of the DNA strands in the telomeric regions, which are detected at the ends of the eukaryotic chromosomes. The repeated nucleotide region is called the telomeres. The telomeres consist of non-coding DNA and block of loss of essential DNA from the chromosome ends. Telomerase is a reverse transcriptase 2, which carries its own RNA molecule that is used as a template during the telomerase elongation, which is shortened after each and every replication cycle. In 1973, Alex Olenikov first predicted existence of a compensatory mechanism for the telomere shortening. He also suggested the aging hypothesis of telomere and the telomeres connections to cancer. In 1984, Carol Grieder and Elizabeth Blackburn discovered the telomerase. The purpose of telomeres and telomerase in the cell aging and cancer demonstrated by the scientists of the biotechnology company Geron with the cloning of the RNA molecule and the catalytic constituents of the human telomerase and the production of a PCR based assay for the telomerase activity known as the TRAP assay, admitting for a survey of the telomerase activity in various types of cancer. The cleavage of phosphodiester bonds is catalyzed by the DNA nucleases. These enzymes play an important role in many DNA repair processes that involve in DNA replication, nucleotide excision repair, mismatch repair, base excision repair and double strand break repair. New nucleases that involved in various DNA repair processes have been reported recently. Severe immunodeficiency or genetic instability is caused by the defects of these nucleases. Thus, structural biology on many nuclease actions is needed for the clarification of the molecular mechanism of the complex DNA repair machinery. 3D structural information of the nucleases is also rapidly gathering, further providing some important insights into the molecular architectures along with the cleavage mechanisms and DNA recognition. Two types of DNA nucleases are present. They are exonuclease and endonuclease. Proofreading is the process of removing the incorrectly matched bases from the newly formed daughter strand. Mismatch repair is a dissimilar process and it takes place later if the proofreading fails. Exonucleases, that is exo 1 and exo 7, involved in the mismatch repair system. This DNA nuclease related with the replication is 3' exonuclease. The enzyme is not a separate enzyme, but it is a part of the DNA polymerase enzyme. Proofreading only deletes the recent errors in the DNA replication. Topoisomerases are enzymes that determine the overwinding or underwinding of the DNA molecule. The winding problem of the DNA molecule develops because of the intertwined nature of its double helical structure. For example, during the DNA replication, the DNA molecule becomes overwound in front of a replication fork. If it is left intense, this tension would gradually stop the DNA replication process. To help these types of the topological problems that are being caused by the double helix, topoisomerases bind to either double stranded DNA or single stranded and cut down the phosphate backbone of the DNA molecule. This mediate break allows the DNA molecule to be unwound or untangled, and at the end of these processes the DNA backbone is sealed off again. As the overall connectivity and chemical composition of the DNA molecule do not change, the untangled and tangled DNA molecules are the chemical isomers that differ only in their global topology. All forms of DNA replication is carried out by the DNA polymerases. Generally, the synthesis of new strands cannot be initiated by the DNA polymerases. They can only extend an existing RNA or DNA strand that is paired with the template strand. To start the synthesis process, a short fragment of RNA known as primer must be developed and paired with the template DNA strand. The DNA polymerase enzyme produces a new strand of DNA molecule by extending the 3' end of an already present nucleotide chain, adding together new nucleotide that matches the template strand one at a time through the creation of the phosphodiester bonds. The energy that is needed for this process of the DNA polymerization generally comes from the hydrolysis of the high energy phosphate bonds which lies between the 3 phosphates that has been attached to each of the unincorporated nucleotide triphosphates. They are nothing but the NTPs. When a nucleotide is added to a growing DNA strand molecule, the phosphodiester bond formed in between the proximal phosphates of the nucleotide towards the growing chain is helped by the hydrolysis of a high energy phosphate bond with a dismissal of the 2 distal phosphates as a pyrophosphate. Generally, DNA polymerases are extremely accurate with an intrinsic error rate that is less than 1 mistake for every 10 to the power 7 nucleotides is being added. Additionally, few DNA polymerases also have the ability for proofreading. They can get rid of the nucleotides from the end of a growing strand instead to correct the mismatched bases. Post replication mismatch repair mechanisms start monitoring the DNA molecules for errors that is being capable of characterizing the mismatches in the newly produced DNA strand from the original strand sequence. Altogether, the three said discrimination steps alter the replication fidelity to less than 1 mistake for every 10 to the power 9 nucleotides that is being added. A restriction enzyme or restriction endonuclease can cut the DNA molecule in between the two identified nucleotide sequences known as restriction sites. All restriction enzymes normally make two incisions to cut the DNA molecule, once through each sugar phosphate backbone of the DNA molecule's double helix. The restriction enzymes are normally found in archaea and bacteria and provide a defense mechanism against the invading viruses. Using the restriction process, the restriction enzymes selectively cut the foreign DNA in a prokaryote. During the process, the host DNA is protected using a modification enzyme that alters the prokaryotic DNA molecule and blocks the cleavage. Together, the above two processes form the system known as the restriction modification system. In detail, over 3000 restriction enzymes have been studied and more than 600 of these restriction enzymes are commercially available. These restriction enzymes are often used for the DNA modification in the laboratories and are an essential tool in molecular cloning method. Naturally forming restriction endonucleases are distinguished into 4 groups. They are types 1, 2, 3 and 4. This is based on their enzyme cofactor requirements, composition, the position of their DNA cleavage site that is relative to the target sequence and the nature of their target sequence. All types of enzymes identify a particular short DNA sequence known as recognition sites and takes out the endonucleolytic cleavage of the DNA molecule to give particular fragments with the terminal 5' phosphates. They vary in their cleavage position, recognition sequence, cofactor requirements and subunit composition. Type 1 enzymes cleave at the sites that are remote from the recognition site. It requires both the S-adenosyl L-methionine and ATP to function. Type 1 enzymes are multifunctional proteins with both methylase and restriction activities. Type 2 enzymes cleave within or at short particular distances from the recognition site. Most of the restriction enzymes require magnesium as cofactor. Type 2 enzymes are single function enzymes that are independent of the methylase. Type 3 enzymes cleave at the sites of a short distance from the recognition site. Type 3 enzymes require ATP. S-adenosyl L-methionine induces the reaction but is not needed. This exists as part of a complex with the modification methylase. Type 4 enzymes target the modified DNA molecule. Example, hydroxymethylated, methylated and glucosyl hydroxymethylated DNA. DNA ligase helps the joining of the DNA strands together by catalyzing the phosphodiester bond formation. It plays a vital role in repairing the single strand breaks in the duplex DNA molecule in the living organisms, but some forms such as DNA ligase 4 may particularly repair the double strand breaks. The single strand breaks are generally repaired by the DNA ligase by using the complementary strand of the double helix as a template molecule, with the DNA ligase by creating the final phosphodiester bond to completely repair the DNA molecule. DNA ligase enzyme has some applications in both the DNA repair and the DNA replication. Additionally, the DNA ligase has wide use in molecular biology laboratories for the recombinant DNA experiments. The purified DNA ligase is used to join the DNA molecules together to form recombinant DNA in gene cloning technique. The process of the DNA ligase is to form two covalent phosphodiester bonds between 3-hydroxyl end of one nucleotide and with the 5-phosphate end of another nucleotide. For the ligase reaction, ATP is required, which proceeds in three main steps. Pyrophosphate is released when adenylation of a lysine residue in the active center of the enzyme occurs. Pyrophosphate bond is formed when the AMP to the 5-phosphate is transferred to a donor. A phosphodiester bond is formed between the 5-phosphate end of the donor and the 3-hydroxyl end of the acceptor. LI-LIC gene encodes the Escherichia coli DNA ligase. The energy that is gained by cleaving the NAD nicotinamide adenine dinucleotide to develop the phosphodiester bond is used by the DNA ligase in Escherichia coli. The Escherichia coli DNA ligase does not ligate the blunt ended DNA molecule except under the conditions of molecular crowding along with polyethylene glycol, and it cannot join RNA to DNA effectively. The T4 DNA ligase obtained from the bacteriophage T4 is the most commonly used ligase in laboratory. This ligase can ligate the cohesive ends of DNA molecule, oligonucleotides and also RNA DNA hybrids and RNA molecule, but not the single stranded nucleic acids. The T4 DNA ligase can also ligate the blunt ended DNA molecule with much greater skillfulness than the Escherichia coli DNA ligase. A T4 DNA ligase cannot use NAD and it has an absolute need for the ATP as a cofactor. There are four types of ligase present in mammals. DNA ligase cuts the incipient DNA molecule of the lagging strand after the ribonuclease H has removes the RNA primer from the Okazaki fragments. DNA ligase 3 complexes with DNA repair protein XRCC1 to help in sealing the DNA during the process of the nucleotide ablation repair and the recombinant fragments. Of the all known mammalian DNA ligases, only ligase 3 has been found to be present in mitochondria. DNA ligase 4 complexes with XRCC4. It also catalyzes the final step in the non-homologous end by joining the DNA double strand break repair pathway. DNA ligase 4 is also needed for BDJ recombination, the process that produces diversity in immunoglobulin and T cell receptor loci during the immune system development. Adenosine triphosphate is rather used by the DNA ligase from eukaryotes and few microbes than NAD. Ligase from many thermophilic bacteria have been sequenced and cloned and are readily available for use in ligase amplification reaction commercially due to their thermostable properties. In this session, we discussed about the DNA enzymes, DNA manipulation process, and the types of enzymes, DNA polymerases, the restriction endonucleases and the ligases. Enzymes are larger biological molecules that are responsible for the hundreds and thousands of metabolic processes that maintain life. Enzymes adapt a particular 3D structure and they may also utilize inorganic and organic to help in catalysis. The process of developing two identical copies from one original DNA molecule is known as DNA replication. The enzymes that take part in the DNA replication process are helicase, primase, DNA polymerase 1, 2 and 3, ligase, telomerase, nuclease, topoisomerase, single strand binding proteins, DNA gyrase and primase. The main function of the helicase enzyme is to unwind the DNA. Primase is an enzyme which involves in the DNA replication process by catalyzing the synthesis of an RNA primer. Telomerase enzyme, also known as ribonucleoprotein, that adds the nucleotide repeats to the 3' end of the DNA strands in the telomeric regions which are present at the ends of the eukaryotic chromosomes. The cleavage of phosphodiester bond is catalyzed by DNA nucleases. Two types of DNA nucleases are present. They are exonucleases and endonucleases. Topoisomerases are enzymes that determine the overwinding or underwinding of the DNA molecule. All forms of DNA replication is carried out by the DNA polymerase enzyme. A restriction enzyme or a restriction endonuclease restricts or cuts the DNA molecule at or near particular identified nucleotide sequences known as restriction sites. Ligase that helps the joining of the DNA strands together by catalyzing the phosphodiester bond formation.